Hi, I'm Liz Suter. I'm an assistant professor at Malloy College, and I'm going to show you how to log into the CyVerse environment and use the QIIME2 pipeline in order to analyze Amplicon data. So the first thing you need to do in order to do this is make a CyVerse account. CyVerse is a computational infrastructure funded by NSF for analyzing life science data. And the account is free. And once you have account credentials, you should be able to log in from cyverse.org with those credentials. The other thing you want to be able to do is log into the CyVerse data storage area using an app called Cyberduck. There are other apps that can do this, but the one that is recommended by CyVerse is called Cyberduck. And there, I put a link in the tutorial to CyVerse's how-to for transferring data using Cyberduck. So Cyberduck essentially lets you drag and drop files to and from the CyVerse data store. So once you follow that tutorial, you've downloaded Cyberduck, you log in with your CyVerse credentials. You should see an area that looks like this. So the root and the home of the CyVerse data store looks something like iplant/home/username. So my username is eSuter. And then within the eSuter folder, I have an analyses folder, and then these other folders are ones that I recently made. So if your account is brand new, you'll only see the analyses folder. So again, I put this link on the tutorial, so you should be able to set up Cyberduck in order to connect with the CyVerse data storage area. Once you've logged into the CyVerse website, we're going to use a portion of CyVerse called the discovery environment. So the discovery environment is supposed to be sort of like a visually interactive environment. So instead of sending your data to the supercomputer and then getting results back, the discovery environment is a little more interactive and allows you to use tools like Jupyter Labs or RStudio so that you can see what you're doing and the figures that you're generating while you're doing your analysis. So once you're logged into cyverse.org, you'll see a series of services. If you don't have discovery environment listed there, it should be in the available tab. Just go to the available tab, request access, and then it should appear in my services. So once you have discovery environment there, you want to launch the discovery environment. It takes a few minutes. And the discovery environment, the first time you log in, should look like this. It sort of looks like a computer desktop. And you have 3 buttons over here. One for transferring data called data, one with apps. So these are computational environments that are already preloaded with certain tools that come ready to go that CyVerse has set up for us, and an analyses button, which contains all of your current or recently run analyses. So the first thing we're going to do from here is check on our data store. So whatever you see in your data store folder should match what you are interacting with in Cyberduck. So if you uploaded some files into the CyVerse data store using Cyberduck, you should be able to see those files here. And later, when we do the analysis, this is where this is also where your analyses folders will see your data files. So you want to make sure that they're there. If you have stable internet. Okay, there we go. So we can see my eSuter folder. If I open that up, I should see the same folders that I was seeing in Cyberduck. And so again, the way to interact with this is to drag folders into and out of this area using Cyberduck. So you can see my BVCN folder, my analyses folder, those are all here in my eSuter folder. The next thing we want to do is start a pipeline for using QIIME2. So like I said, discovery environment is really useful because it comes with these environments that already have certain tools loaded into them. So we want to pick one that has QIIME2 loaded into it. So the first thing you want to do is press the analyses button. And this is the area where we can, oops, sorry, I lied. The first thing you want to do is click the apps button. So the apps are all the environments that CyVerse has already prepared for us. And you can start searching for some. So I know that the one that I want to use is called Jupyter Labs QIIME2. So I'm going to start typing Jupyter Labs in order to search for it. Or just Jupyter. And there's a couple of apps that use Jupyter. So you can see a Jupyter Lab QIIME2, a SciPy, and so on. So, and there's another QIIME2 here. So because I've been looking through this a little bit, I know that this Jupyter Lab QIIME2, the first one that pops up here, is the most up-to-date version. So it's using a newer version of QIIME2. This one is using an older version of QIIME2. So I'm going to want to use this newest one. One thing you might want to do is click on this little button and add it to your favorites. So mine says remove from favorites because I already have it in my favorites, but you might want to add it to your favorites so that now whenever you open up your favorite apps here, it'll appear there. So if you click on the name, or if you click on those little triple buttons there, we can launch it. I'm going to click on the name here because I don't want to do quick launch. I actually want to select some parameters when I launch it. So the first thing is we can name our analysis session. So I'm going to keep the name that CyVerse or Discovery Environment has already used. Jupyter Lab QIIME2 2019.10 is the version of QIIME2. Instead of analysis 1, I'm going to call it Happy Belly Test because we're using the data from the Happy Belly Amplicons example. Discovery Environment also puts automatically selects your analysis folder from the data store as the place where to put your analysis files. That's fine with me, so I'm going to leave it as is. Next, I'm going to expand this little input data section. So this is where you tell the analysis session where you're getting your data files from. So you click Browse, and I put my data files in a folder called BVCN in my eSuiter folder using Cyberduck. So I'm going to direct it to this BVCN folder and click OK. The next thing you want to do is select your resource requirements. So for this Amplicon analysis, we are going to use the maximum for each of these. So we're going to use 8 CPU cores, 64 gigabytes for minimum memory, and 512 for minimum disk space. And that's it. So then we click Launch Analysis. So I'm going to close this apps window, close my data folder. Now, up here in the corner in this little alert section, it's telling me that my analysis has started running. So I can access it that way, but the analysis is also going to be available here in my analysis area in this analysis button. So right now I actually have 2 analyses running because I started one before, but the most recent one that I just set up just now is Happy Belly Test. So if I click on this little button here, go to analysis, it will open up my Jupyter Lab, and it takes a little while to launch. Okay, once your analysis has launched, you're inside a Jupyter Lab environment, and this is what it looks like. So you have the option here to make a Python 3 notebook. You also see my BVCN folder where our temporary working space is. And then you also have the option down here for some other types of coding environments like the terminal. So the first thing that you want to do is connect this Jupyter Lab to the data store. And you do that by clicking on this little CyVerse swooshy button here. And you're going to enter your CyVerse credentials again. So my username in this portion and my password, which it saved from last time, and submit. There's one more thing you have to do in order to connect to the data store here. You have to open a terminal window and I initialize. So the command for that is IINIT. And once again, you're going to submit your CyVerse password, which I'm copying from here and pasting it. As you type this, you won't see any text appear. Just type out your password and press enter. And if it shows the next line with no errors, it worked. So this is just like a terminal environment. So if I press LS, it's going to show me my current environment. And it shows that BBCN folder. These other folders are just log files that will automatically appear. So one more thing I wanted to show about this environment in particular in CyVerse is that using the terminal, you can use the typical commands. So ls pwd shows you your present working directory. You can do cd, mkdir, those types of commands. And those are all interacting with this temporary environment where we directed the discovery environment to pull its files from. But we're also working in the second file system, which is the more permanent file system in the data store. And that's where we put our files using Cyberduck. So we can also interact with that file system using I commands. So whereas ls shows us the files that are present in the current temporary directory, ILS, the I command for ls, shows us the file system in the data store where we're located. And you can do the same thing with PWD. So PWD shows us that we are in work folder in the temporary environment. If I do IPWD, that shows us that we're in the eSuiter folder in my data store area of CyVerse. And one last thing I want to show you about I commands is that you can use them to move files from the temporary Jupyter Labs environment to the permanent data store and vice versa. And you do that with the I commands IPUT and IGET. So if I just really briefly type IPUT -H for the help menu, it'll tell you all the parameters you can do for IPUT, but essentially this is for storing a file into iRods, which is that data store, which is the area where my permanent files are, the data store in CyVerse. So that's would be if I want to move a file from my temporary environment to the data store. And if I try IGET, IGET is to get data objects or collections from iRods either to the specified local area or to the current working directory. So you can do this really basic file transfer in the terminal using IPUT and IGET to interact with your CyVerse data store. Another way of doing that is to go back to the Discovery Environment tab or portal. And if we open the analyses window, you'll see all of my current working analyses or running analyses and completed analyses. So if you're all done with your analysis and you want to end it, you can press this button and press complete and save outputs. And that will end the analysis session and send all of your files to this data store in the analysis folder. And it goes into the analysis folder because when I started this analysis, that's where I directed it to go. You could have chosen a different place in your CyVerse data store if you wanted to. While I'm here, I want to show you something else. The discovery environment allows you to launch an analysis for a set amount of time. It's usually about 3 days. So you can see the start date for this and that it's currently running. If I was coming on to towards the end of the time limit of the 3 days and I wasn't ready yet, I could extend the time limit by pressing this little hourglass. And so it tells me this session is due to end or expire on May 21st. Would you like to extend it? And it should extend it a couple more days for you if you say yes. If you don't extend it and the session runs out, CyVerse will actually automatically send all of the files that you generated in this temporary environment into that analysis folder anyway, including any workbook or notebook files that you might have generated that are here temporarily. So it's actually really helpful feature so that you can't accidentally lose anything in case you get distracted and start working on another project. The last thing I want to do in this video is show you how to launch a notebook. So we go back to the launcher by pressing this little cross, and we're going to launch a Python 3 notebook. And we're going to run, this is because we're running our QIIME2 in a Python 3 notebook. QIIME2 actually runs in Bash, so you could run it from the terminal window like we did here. Like we were running commands here. We could run the QIIME commands here as well, but we want to use this Python 3 notebook because, and this is a special feature of this CyVerse, of using the CyVerse environment, because this notebook comes preloaded with a feature that allows us to visualize the outputs of QIIME2 directly in the notebook as we're doing our analysis. And the only tricky thing here is because it's a Python 3 notebook, we have to be using Python. So Python has some commands that are similar to Bash. So if in my notebook cell I write ls, it recognizes that as a Python command. But ls can also be a Bash command. And I can tell the Python notebook that I want to run a Bash command by putting an exclamation point, ls, and now it runs it as Bash instead of as Python. So throughout the QIIME2 tutorial, we'll be going back and forth between running Bash commands, which most of the QIIME2 commands are written to be that way. So we need to use the exclamation point, or occasionally we'll use a normal Python command, and that will be mostly when we're looking at the data visualizations. So that's it. In the next video, I will actually jump right into QIIME2 from this environment.